So many of you in the audience have probably heard of a biotechnology called CRISPR. This biotechnology is known to be revolutionizing life science and medicine. Over the last decade, many magazines and top-tier academic journals have highlighted CRISPR as this breakthrough technology that is actually allowing us to modify life as we know it. CRISPR is powerful because first of all, it works. It can edit cells, and it works for almost everyone. You don't have to have a lot of training to make it work. And the other reason that it's powerful is that it's inexpensive. The fact that this transformational tool has been so impactful led to the awarding of the 2020 Nobel Prize to the two scientists who discovered this technology, Emmanuelle Charpentier and Jennifer Doudna. But I want to note that these scientists, when they made the discovery of this CRISPR tool, they weren't actually looking for a new genome editor. What they were looking for was just an understanding of a war that has been going on since life began. This conflict is the conflict between bacteria, which in this electron micrograph is colored as blue, and viruses that kill bacteria, which are these T4 bacteriophages, which are colored green. T bacteriophages that try to kill bacteria and need them as hosts, they outnumber bacteria at least 10 to 1. And bacteria have in turn evolved immune systems to protect themselves from this pervasive threat. One of these immune systems was discovered almost a decade ago, and it's called CRISPR. Now, again, most of you think of this as a tool, but CRISPR actually is an acronym that is shown there on the right of the slide, clustered regularly interspaced short palindromic repeats. It's quite a mouthful. What it's trying to do is describe something that was observed in the genome of bacteria in 1987. We have repeats, which I'm showing diagrammatically there on that little train track, which is meant to be the genome of a bacteria. The squares or the diamonds are the repeats. They're about 30 bases. And then they're interspaced with other sequence that is unique from the repeats and from each other. So although this repetitive element had been observed in bacteria for a long time, the biological significance of it was not known for many years, up until actually 2005, when phylogeneticists discovered that these unique repeats weren't actually unique at all, but they were identical to the same sequence of viruses and plasmids that infect these microbes. This single discovery led to what my mentor called an idea grenade. These scientists thought, why does it have DNA from the viruses that are infecting it? Maybe this is a memory, and perhaps bacteria can adapt and then protect themselves for a future attack. The scientific community actually didn't like this idea and rejected it at first, but it is actually true. This is what bacteria do, and that's what these CRISPR systems are. They're adaptive immune systems. And really quick, I just want to walk through how they work. So notice that there's some genes next to the CRISPR that I diagrammed there. These genes encode for proteins, and some of these proteins are involved in acquiring new sequences from viruses. Maybe you didn't notice the little virus on the corner of my cell there, but it's trying to put in its DNA. It grabs DNA and integrates it into the CRISPR. This memory is then transcribed into a single-stranded RNA, and this long RNA is then processed into a library of small RNAs. Each small RNA containing a sequence that was derived from and complementary to a virus. Other proteins combine with this RNA, forming a ribonucleoprotein complex, and the RNA is used as a homing signal to find complementary nucleic acid. Once it binds, the nucleic acid of the virus is then cut. And this protects the bacteria from that invading virus. Okay, so these three stages, acquisition, biogenesis, and interference, these are found in every CRISPR bacterial immune system. But what I'm going to show you next is the diversity of CRISPR systems. I know that you can't see everything on this slide, but what I hope you can see is that there are 6 distinct types, and then there are at least 43 subtypes of CRISPR systems that have been discovered to date, and we expect to discover more. Each of these types has distinct genes, and that's what you're seeing here with the little arrows. And we, as a field, and I certainly as a person, hypothesize that these distinctions will allow for discovery of new technologies. We think they'll all be guided by RNA, but the way that they will protect or engage with foreign nucleic acid could be distinct. So the CRISPR system that you've all been probably, the folks, if you've heard of CRISPR, the CRISPR system that you probably have been aware of is this one here that I'm highlighting here. So just one of 43. It's called Cas9. And usually when I say Cas9, heads nod. Oh yeah, I've heard of Cas9. So let me just really quickly walk you through how Cas9 works. So this is the operon of the traditional Cas9 that most labs and even doctors are using out there in the field. So Cas9 is a protein, and here we have a gene that encodes for that protein, and then we have a CRISPR next door. The CRISPR encodes for an RNA, and then there's another RNA called the transactivating RNA that forms a duplex that is then processed into that library of small RNA. The protein itself is then made and combines with that RNA to make an RNA-guided surveillance complex, or a ribonucleoprotein complex. In a cell, what this would do is use the guide to find complementary DNA. Once it finds its complement, Cas9 binds and undergoes a conformational change. That conformational change moves parts of the protein into position to cut the double-stranded DNA, leaving a lesion. Now, in this case, I'm showing you what happens in bacteria, where Cas9 breaks the DNA of the invading bacteriophage, and that's a win for this particular bacterium. But let's go back to Manuel and Jennifer. They started doing microbiology, and they started finding out all the key pieces to this, and they realized that they had a very powerful tool. If they could make a double-stranded break in DNA on purpose in human cells, they could perhaps edit the code of life. So they actually were successful with this. And let's talk about how this happens. So 1, we can make protein in the lab. And 2, we can make RNA in the lab. And we can change the sequence of the RNA pretty easily. And so the other problem that was at hand is we just had to get the Cas9 protein into the nucleus, and that could be done by adding a small amino acid chain called the nuclear localization signal to get it across the nuclear membrane. This allowed Cas9 to enter the nucleus, the guide RNA got it to the DNA, and a cut was made. That's all that CRISPR does, is it makes, or at least CRISPR-Cas9, it makes this double-stranded break. Then scientists rely on the machinery within the cell to repair that break to do the actual gene editing. So there's 2 processes that can happen here. The first is that a process called non-homologous end joining inserts or deletes bases and creates what we call an indel, and that effectively knocks out a gene. The other is that you provide some sort of a donor, and you're relying on homology on the flanks of the lesion with your donor so that it will insert a new sequence. We call that homology-directed repair. This allows for editing that knocks out genes or adds in DNA sequence where the scientists may want. And so this has already led to all kinds of cool applications in both agriculture and medicine. I do not like browning mushrooms. And so imagine just knock out that enzyme that makes them brown. Or hey, I'm a cattleman and I don't like the horns. Well, let's just edit the genome and knock out that gene. You can see where this is going. But there's also been therapeutics in humans. One of the most successful is what I'm showing here. These 2 individuals were cured of the symptoms of sickle cell anemia. Sickle cell anemia is a blood disease, and that makes it easy to treat with gene editing because you can actually take the stem cells out of the bone marrow, manipulate them genetically, and then put them back in. And that's how CRISPR has helped these 2 individuals. Some crazy ideas that are out there now that used to be science fiction. but because we have this gene editing tool, are de-extincting animals like the mammoth, reversing aging, creating plants that can detect explosives, and creating pigs that are growing humanized organs. This used to be science fiction, as I said, but now it's on the table. So with all of that, this is what's going on with Cas9. But I told you with my title that I was going to tell you what we're doing at Utah State to discover new CRISPR-based tools. And so what we're mining is this diversity. When I started my lab just 7 years ago, I realized that many of these systems had not been studied. So I started looking into these, what were then newly discovered systems. And I'm going to tell you a story about one of them. Before I get to all of the details, I wanted to show you a movie. I like movies, and I hope it plays. Maybe it doesn't. Oh, there it goes. Okay, great. So this is a three-dimensional structure that we determined with a method called cryo-electron microscopy. One of our foundations is what we call structural biology. We believe that if we can see what's going on, that'll help us understand what's going on. And so we used cryo-EM to capture a machine, of a CRISPR machine, in the process of interacting with a nucleic acid. We use other strategies like microbiology and biochemistry to answer questions about these bacterial immune systems. And so now what I'm going to do is, instead of trying to teach you all with the data that we generated, I'm going to use a schematic just like I did with Cas9 to show you what we have recently learned about a different system. So the system I'm going to talk to you about is called Cas12a2. Cas12a2 is found on the genome of an organism that lives in a cave called Sulphuricurvum. The genome has a gene that encodes for the protein, and then you have the CRISPR that encodes for an RNA. They combined very similarly to Cas9 to form a ribonucleoprotein complex. Our first biochemical experiments that we did in the lab, we thought that this would bind double-stranded DNA, just like Cas9. We tried and tried and tried, and we always came up with nothing. I often thought it was my students' fault, but it actually wasn't. We learned later that this instead binds single-stranded RNA. And when it binds a complementary single-stranded RNA, the protein undergoes a huge conformational change. The way I like to say it is it gives the RNA, that's the target, a big hug. And in doing so, it opens up a different domain so that it can be accessible to degrade nucleic acid. This nucleic acid degrading domain is what we call indiscriminate. It does not care what it degrades, and it starts degrading double-stranded DNA, which really effectively destroys the phage, or the virus. But what also happens is it also starts degrading the chromosomal DNA. And as you can imagine, that would probably not be good for that particular cell, resulting in cell death. Now, overall, this is good, though, for the colony, and it makes sense why it'd be evolutionarily conserved, because bacteria that get infected and then stop the infection protect the neighbors in the bacterial colony. So where are we going for this? We made this discovery that instead of making a single cut in double-stranded DNA, Cas12a2 binds RNA and then kills cells. What can we do with this? Well, we have a lot of ideas that we're now pursuing, and this is at Utah State and through several collaborators, including collaborators at University of Utah. First of all, we think we can do some pretty cool RNA diagnostics. We can use this to detect RNAs from viruses or any RNAs that are in the environment. The coolest experiment that we can come up with is something like selective killing, where we have a pool of cells and cells that are genetically distinct at the RNA level. We kill the ones that have the RNA and leave the others. This could be very powerful in something like cancer treatment, where cancer has RNA that's distinct and the healthy tissue doesn't have that RNA. We imagine a chemotherapeutic where you only kill the cancer and you do not kill any healthy cells. Obviously, we could shape microbial communities. And back to that same idea, and this is where I think there will be a powerful tool, maybe where it'd be used the most, is imagine someone's trying to make a gene edit, and this could come in and kill any cell that was not edited, enriching for editing in cells that are maybe difficult to edit with CRISPR-Cas9. So I just want to finish by saying this arms race that's happening between viruses and bacteria is a huge treasure trove of technology, I think. And we've really only scratched the surface. I have a team of excellent students at Utah State University. They're smart, and they're happy, and they're fun, and I'm just really excited to work with them. And we anticipate that this strategy we're using will lead to future discoveries. And I will just say one thing that I'm really advocate for here at BioHive. I think that we should be talking more with the students and the amazing industry that's happening here at in Utah, Salt Lake City, trying to make internships happen, because I see these really smart students and I see these really cool innovations, and I just would love to see more happening because I think it's going to lead to amazing innovations. With that, I'd just like to thank my many collaborators, my students, and especially the funding that I've received through NIH. Thank you so much.